Oh, I love it. I mean, I've been here 3 times now, and every time I come here, I see what has been done. I think the excitement here is something that other universities should pay attention to. There is something of a hunger for information. I think I made a point earlier in my lecture when I started that these kids make me feel young. I think it's wonderful, and the number of questions that were asked at the end. I mean, even finishing the second, I could have easily gone on for another hour or so if I wanted to. I absolutely love it. I think it's uplifting. So there were 2 things that we discussed in my lectures. I don't call them lectures, I call them discussions. The first one was on how you secrete molecules, vital molecules, that are too big to fit into your standard, well-known transport intermediates. Yeah? So one was about the size matters, and the second one was about quantities. So, for physiology, you need to secrete these vital molecules in the right quantities, and then you have to worry about the size. There are molecules that are too big to fit into the textbook material. These are long-standing questions in our field, and we have made these questions amenable to molecular analysis, and that's what I presented. The ability to do molecular biology in general, the ability to change at will, mutagenize, and generate new forms of proteins, to be able to rewire cells, are the kind of technologies that are really helping, not only to address questions that were not amenable to analysis before. You can do experiments in human cells, you can derive one cell type from another. I mean, this whole business of the discoveries that have to do with CRISPR-based knock-ins and deletions and changing cellular pathways, engineering cellular pathways, to be able to analyze massive amounts of data. This has all made biology easy to address. You can ask questions that you could not even dream of before. So, many technologies have made it possible for us to be able to visualize individual molecules, to do experiments in living cells, to be able to create 3D structures of a tissue and be able to analyze a process, not just in isolation in a cell, but in a tissue. I think are really helping us in terms of understanding the basic principles, and then be able to apply that knowledge to address human pathologies. This group of people, my God, they were asking me afterwards advice on PhDs and this and that, and I was saying, come to my lab. So they are really aggressive in the sense that they want to make a difference. Yeah, it's a tough course, and they could easily be whiling away their time doing nothing. So I think there is a level of dedication that is touching.